Hi everyone. So today we are at Warwick, the university that I'm doing a PhD in. Our guest speaker today will be someone from my lab. We share the same lab space, but we are not from the same lab group. He will be the first non-Thai speakers that's gonna be my guest speaker. So I will let him introduce himself. Hello.
Hello. Hiya. Well, first, thank you for including me in the researcher series. My name is Gerald. I'm from Malaysia, specifically East Malaysia, from a small village there. So, a little bit about myself. I did all my schooling in Malaysia up until my bachelor's degree. And then I did a master's in Europe under the program Erasmus Mundus, joint master's degree. So that involves going to different countries every semester. I also work with the Malaysian government for a bit. And then finally, it has always been my dream to do a PhD. So finally I'm here in Warwick, yes.
I think you once showed me your photo, like underwater. What did you do there?
Oh, okay. So when I was working, I was posted in several divisions. So I worked in a lot of areas. So one of that is I did a lot of diving. So diving in terms of conservation, cleaning up ghost nets. So ghost nets are fishing nets that are left or they detached from the fishing vessel, or fixing anchors for ships to stow. Yeah, among all those other things.
Oh, that's so cool. So how come you got your PhD here? So which funding did you get?
So I spent a lot of time, so several years applying for a PhD. So I applied in Malaysia, in Thailand, in South America, also in Europe. So it's very hard for international students to get opportunities overseas. Finally, from my master's, I have some reference from my supervisors. So he connected me to different people. Finally, I saw this advertisement. So my funding is funded by the European Research Council. So this is a grant where professors or PIs, they apply, and then they get this money, and then they can hire postdocs or PhD students. So that's where my funding comes from.
I see. Just now you mentioned Thailand. I think you also mentioned that you once collaborate with people in Thailand or something.
Yeah. I did several projects with FAO of the UN, also with NACA. So NACA is a regional body that runs a program for aquaculture. So its office is in Bangkok. It's inside Kaset University.
Oh, that's it.
Yeah, so from this program, so I met a lot of people from different countries, and some of them are from Thailand. So yeah, so we are friends.
Nice. So now we will be talking about your PhD project. Yeah. So what's your project about?
So the grant that I'm involved in is called VERFIX. So the overall aim is to understand how viruses in the ocean impact the carbon cycle in the atmosphere. So we have a lot of postdocs. I'm the only PhD student. So my role is, specifically my PhD project, is to understand how does the viruses infect cyanobacteria. So cyanobacteria are bacteria that photosynthesize. So they produce most of the oxygen that we consume in the world, and they also are the primary producers, meaning they fix carbon and they turn the carbon into, to put it simply, sugar. So for the energy for other organisms to eat them. So you have a transfer of energy up the food chain. So then there's these viruses or bacteriophages. So what they do is they infect the cyanobacteria, and they have this group of genes inside them which they use to manipulate the bacteria. I'm trying to understand, how do they obtain these genes? Are they different in different ocean areas? So what I do is I try to delete these genes and see how do they affect. And at the same time, I also try to understand how does the cyanobacteria fight against the viral infection. Specifically, I'm trying to understand how it modifies the outside, the surface of its cell.
Yeah.
Yeah, to fight against virus infection. The impact of my research would be we would understand more the role of microorganisms in the ocean, how it affects the climate, the atmosphere, and in my case, the carbon. So one of the genes we are working in, we suspect it's involved in stopping carbon fixation in the world. So on a larger scale, because the Earth is covered by 70% of the ocean, so if this gene is responsible, then it has a global impact.
Yeah.
So that's the main thing, I think.
Now we will show where Gerald's work. So this is the C126 lab. I'm also working here.
We keep our cyanobacteria that we work on. So all these are our shaking incubators.
Yeah.
So because our cyanobacteria, they photosynthesize, so we need light. So we can't keep in a normal incubator.
Yeah.
So we culture different types of cyanobacteria. So they come in different colors. So for example, they have orange, you have pink, you can see, and you have green. So they all have different colors because they have different pigments inside their cell wall that gives out different colors. But all of these pigments are very important for photosynthesis.
Yeah.
Generally, they multiply once a day. So we need to wait around 2 weeks to have a volume or cell number that we can work with.
Yeah.
So I'm going to show you. So this is one of the places we keep our cultures. We try to keep our cultures very acidic as possible. This is one of the rooms where we keep our stock. So we have a place where we put working cultures and we have a place where we put our stocks.
So this is the 23 degrees.
That's right. So as I've explained earlier, we have lots of different types of cyanobacteria. So most of them are marine strains, so it means they live in the ocean. Most of them belong to the genus Synechococcus. We have also Prochlorococcus. So these two genera are the most abundant cyanobacteria in the ocean. So they are very, very important. They are everywhere. They are in the most nutrient depleted areas, and they exist from coastal to open ocean. They are everywhere. They're very, very important in terms of producing oxygen and also fixing carbon in the atmosphere. And this is the non-mutant, so wild type we call. So we deleted a gene that we suspect controls the cell surface modification. So what happened? The cells, this was up at the same time. So the wild type, they can, we say, spread out around the media, so they can float. While the mutants, they all sink.
Yeah.
So
they're all at the bottom of the.
That's right. Yeah. So we don't know exactly why. We only know that it sinks. So that's one of the things that we are investigating.
Do you have any plans after this, like after you finish?
I have some working experience before I coming here, but I do love research.
Yeah.
Because every day I wake up, I don't feel like it's a job that I hate or I don't want to go, or a job that I have to do because I need money. It's something that I really look forward to every morning. So if I have a choice, if I can choose, I would like to stay in research. Yeah.
That's so true. Every time I came to uni, even on the weekend, I met him. Yeah. So now you are in your third year. So you have 1 more year or?
Yeah, so the funding gives me 4 years. Yeah.
How do you like Warwick so far and your research group and everything?
Yeah, I love my research group. They are very good. I also love the people outside my research group. Everyone has been kind here to me. We joke around a lot. We talk a lot. Warwick has been my opinion, very good to me. It's a nice campus, it's a nice school. Funding is good, resource is good, so I have no complaints.
Yeah.
It's been amazing.
What about the UK? Do you miss home, or your family also here with you, right?
Yeah, my family is here with me. Do I miss home sometimes? Yeah, but the UK is also amazing.
Yeah.
I like the weather. It's not as hot. In Malaysia, it's very hot. In terms of food, we can cook.
Yeah.
So it's alright. Yeah.
Thank you, Gerald, for being my guest speaker for this episode.
Yeah, I'm really happy. I've been following Sopida's the series. It's really good. I really love it. It's very compact, concise, and I'm really happy to be a part of it. I hope if anyone is interested can reach out. We can collaborate or just network.
Yeah. So you can contact him. Yeah, thank you very much, and I'll see you next episode. Bye.
Bye.